Okay, so welcome biofans. Once again, we're going to be talking about the one thing about transcription that we never really got a chance to talk about before, which is how do we turn genes on, right? This is actually going to be step one in our discussion. This lecture will be talking about bacterial genes. In the next lecture, we're going to be kicking things up a notch, maybe 10 notches when it comes to complexity, because we'll be talking about eukaryotic cells. Bacterial genes are a little bit different because they've got the structure, the operon structure. So we'll be learning about the lac operon, which is required in order for a bacterial cell to digest, absorb, and use lactose, the sugar. And we'll also talk a little bit about the operon, the tryptophan operon, which the bacteria needs in order to activate, when the bacterium activates that operon, it's able to make the enzymes which are required in order to make the amino acid tryptophan. Okay? And that's what we're going to be getting to today. There's only one section in the textbook that you need to worry about, chapter 11, part 1. And again, a couple of things I just want to have you keep in the back of your mind. We're just learning the basic nuts and bolts about how turning genes on, turning genes off can work, and how it does work in the case of bacteria. We're going to need to stretch this out quite a ways when we talk about the way in which we achieve the same thing in eukaryotic cells. All right? Okay, and just to give you a little bit of a pep talk, there may be some times in which genes are not needed. Okay, and if the gene is not needed, then we could sometimes just shut them off. Right, that's called repression. It's like we've got we don't have to be expressing those genes. If we don't need them, we can shut them off. We don't have to. There are other genes that are that might normally be in a repressed state, and we have to de-repress them. In other words, we have to induce their activity. Transcription is going to be induced in the case of an inducible operon. Okay? And so in the case of a lac operon, lactose operon, that would be an example of an inducible operon, whereas the trip operon is one that will be turning off during the times in which we don't need to produce tryptophan, right? And so keep in mind, I mean, we have a ton of genes. The bacteria also, even the bacteria are much more simple than we are. They've got a ton of genes, and they don't need to be expressing all those genes at all times. They need to be able to economize. It would be a complete waste for a bacterium to produce the enzymes required to absorb and hydrolyze and modify so that they can be used. Lactose is the food. If you don't need to digest lactose, then it makes no sense for you to produce the enzymes required to break down lactose, because you're much better off using those resources for something else, right? And so, yeah, I think I want you to fully take ownership of the idea that yes, we need to turn this gene on. Otherwise, it needs to be kept in the off position. Yes, we need to be able to turn these genes off. Otherwise, it would probably be left in the on position, right? It's like there might be some genes that would be inherently off, turn them on. Some genes that might be inherently on, turn them off. It's not quite that easy, but that's a good place for us to start. Okay. Now, this is an important concept. The differences between the bacterial genome in the way that it's organized and structured as opposed to the eukaryotic cells. We've already talked about this once. We said that bacteria have circular DNA. Should be an I there. So you know that it's in a big circle. It doubles up on itself, whereas in eukaryotes, they're on different chromosomes, linear pieces of DNA. And yeah, that DNA is complexed with things like histone proteins, so we get the material chromatin, all these things we talked about before. Maybe these are the same chromosome, maybe these are the same chromosome, or homologous chromosomes, should I say. But the differences in the structure, we've noted them before. Here we're talking about the organization of genes that are needed together. For example, right, in order to use lactose, lactose is, anybody? It's a disaccharide. Might not have been the first thing that came to your mind. It's also the same as milk sugar. And yeah, for an E. coli cell, lactose is an important food resource sometimes, but not others. Okay? Most E. coli living in a lactose intolerant human being would probably never encounter lactose because the human is behaviorally conditioned not to consume lactose-containing products, right? You get oat milk ice cream sandwiches instead of regular ice cream sandwiches, right? I mean, if you are conditioned to avoid lactose, then the E. coli cells that are living in your gut never encounter lactose, and to produce the enzymes that are required be kind of a waste, right? To use lactose, we need to have 3 gene products. We need to have protein A, protein Y, and protein Z. I'm actually going to be using letters that are appropriate. Yeah, and you were expecting me to say A, B, and C, but no, I'm going to tell you that it's proteins A, Y, and Z, right? One of them is called beta-galactosidase because it converts a galactose into a glucose. Another one is lactose permease because it allows the lactose to enter the cell. And the other one is called beta-galactosidase is the one that cleaves lactose into 2 little fragments. The other one is lactose transacetylase, which converts the galactose into glucose. Yeah, I know what they are because I've taken the essential biology classes, but you don't have to know what they are. All you have to know to realize is that these are proteins that are needed when lactose is present and the bacterial cell needs to use lactose as a food resource, but we don't need these proteins at all under circumstances when the lactose is there, right? Okay, so what we do in the case of E. coli cells, we have this thing called an operon, right? This is the way that genes are organized in bacterial cells. So in the operon, you'll actually have the 3 genes, right? This will be the gene for lac-Z. This will be the gene for lac-Y. This will be the gene for lac-A, okay? And they're going to be, they're inside of this chromosome. These are the 3 genes that are there. They're going to be there inside the chromosome in their designated spot. There's lac-Z, there's lac-Y, there's lac-A, right? At the very top of this, on the upstream side of lac-Z, you've got a control region. Control DNA, which is going to determine whether or not the operon's genes are going to be transcribed. Now, once we transcribe this, once we start producing RNA transcript, let's say there's a RNA polymerase. Once we've made a copy, and here's the RNA molecule coming off of it, right? So this might be the gene for lac-Z. This might be the gene for lac-Y, and lac-A is almost there. We're producing only one transcript. It's like we're not making 3 separate messenger RNA molecules. We're getting one messenger RNA, even though there are 3 genes, and we basically get one long polypeptide, right? The polypeptide that forms, here's the ribosome. Here's the polypeptide that gets produced. The polypeptide is produced, and then we have to cut the polypeptide into 3 parts, right? And then the 3 parts are going to form the enzyme for lactose beta-galactosidase. The next one is going to be forming the membrane protein lactose permease, and this one is going to be the transacetylase. Okay, so in other words, we're getting three, this is our Z protein, Y protein, and A protein. We're getting all three at the same time. All we have to do is to to turn on the operon. We make an RNA transcript, and that one RNA transcript gives us three gene products. Okay? And this is what we mean by the operon structure, right? We're taking genes that are functionally related to each other that you all need together, right? There's no point in producing the A protein without the Y and the Z. Okay, without the Y and the Z, there would be no point to having lac A, right? And without the lac A, there's no point to having a lac Y and the Z. You need all three together, and so you turn all three together, turn on all three together at the same time, which kind of makes perfect sense, right? Our cells don't do it that way, right? If a eukaryotic cell needs to have three gene products, A, Y, and Z, it'll control the activity of the A gene and the B gene and the Y gene and the Z gene all independently of each other. And we would have to produce one RNA transcript over here, another RNA transcript over there, another RNA transcript over there to achieve the same thing, right? In other words, it's like bacteria are doing things far more economically compared to the way that our cells are going to be doing things. Don't worry about this too much. We're going to be coming back to that in the next lecture, right? Right now, I want you to get the idea that what the E. coli cell has to do, this is Mr. E. coli, Mr. E. coli cell has to do now that lactose is present, it needs to turn on these genes in order to get uh, in order to get the gene products that are required. You need to turn it on, and that's the effectively what we'll be talking about next. Okay, and so at this point, I'm just going to point this out to you that we need to focus on the control DNA, this part here. Okay, because we need to make sure that we activate. If we turn things on here, then that's it. Now, we're going to be making the RNA transcript. We'll be translating the RNA transcript many times. We'll be getting the gene products all at the same time. So one switch is going to result in everything else cascading. And so we need to focus what's going on right there at that one header. Right? And I'm going to draw this on the board. Hopefully you'll get to you'll already be familiar with this because you've read about it in the chapter. And so in this control region, okay, this is the control region. Let's say that over here is the lac Z. Okay? And the lac Z will continue on to lac Y and lac A. Okay? So we need to turn it on. But we need to think about who's going to be working with this. We have to have that RNA polymerase. Remember, I kind of drew it like a bullet before. RNA pol. Stands for RNA polymerase. That's the one that's going to be doing the transcribing. It's going to be making new molecules. It's going to be stringing together the RNA nucleotides to make the RNA molecule that will become translated, right? So the RNA polymerase is looking for a region called a promoter. Okay. And by the promoter, what I mean is a special area on the control region that is required to bring in the RNA polymerase and initiate the process of transcription. When RNA pol lands there, okay, it lands. If it were to do that thing and land right here, RNA pol, at that point, it could move in this direction and to start the transcription. We'll be making our molecules in the 5' to 3' direction, and it'll be reading the DNA in the structural region. Will start reading right there. But that's only possible if we are free, okay? Because also on the promoter is part of the control region. because also on the control region, between the promoter and the place where the LAC-Z actually begins, where you need to start reading, is the operator. Operator, right? And so the operator region is going to be over here, and that's we're going to be having the functionality of a repressor. And so the repressor is going to bind to the operator, and oops, let's see, I need to do this with an eraser. So I'm going to draw this in here. Okay, so I'm actually going to draw the repressor down here. Oops, I'll undo. Make that a pen again. We'll draw the repressor down here. It's attached to the operator. And what this functionally does is it blocks the RNA polymerase from making it past this point. Okay? And so with the repressor bound, you're going to be preventing the RNA from getting across that this boundary. We're not going to have, the repressor is bound. The operator, repressor effectively blocks. Transcription from occurring. Okay? In other words, it's repressing, it's turning the gene on, off. Yeah, I told you that the LAC operon is an inducible one. And so we induce activity of the LAC operon by de-repressing, right? Inducing, okay, sorry for the messy handwriting, is going to here mean the same as de-repressing. Okay, so what we need to do is we need to convert the repressor into a non-binding repressor. And we do that with the addition of the, in chemistry, we call it a ligand, the thing that binds the repressor. The ligand that comes in here, binds the repressor, is lactose, right? L, okay? I made it round because it'll fit into the round hole. And so the lactose is going to attach to the repressor at this other site. It's actually allolactose, but don't worry about that. Basically, lactose is coming in, binding to the repressor like this. Right? And once it binds to the repressor changes its shape. And let me do that, do another erasing thing. So here's that lactose again. And now we're bound to the repressor, but now the repressor is attaching like this. The repressor in its new shape is not going to be bound to the operator. It falls off, and now the promoter can do its thing. It can chug along past the operator, and it can get down here to the LAC-Z and start producing the RNA molecule. The RNA is going to be coming off, which is going to be a copy of what the gene that's there in the LAC operon. Right? And so again, at that point, the ribosome is going to be coming in, moving this direction, will be producing the polypeptide. It's going to become the first part of the polypeptide that will eventually become the beta-galactosidase, one of the enzymes that's there. Okay. Yeah. Go ahead and review this. This should be it should be a review already because you've read about this in the textbook. I just wanted you to have a chance to hear it from me so that you, I guess, know that the textbook is not lying to you. Okay. The other thing that you want to think about at this point is this, right? How does this all make sense? This all makes perfect sense if you think about it for like more than a couple of minutes, right? When do when you want off? Well, what do you say? Yeah, we want it off when there's no lactose. Right? And so as long as there's no lactose, this repressor will be bound to the operator, and you won't be getting any transcription, and you won't be getting any production of these enzymes, which are completely not needed if we don't have lactose around. Okay? When is on good? A fancier word there. When is on desirable, right? Well, yeah, we want to turn the lac operon on when lactose is present. So it makes absolutely, completely, totally perfect sense for lactose to be the thing that induces or de-represses this operon. Yeah, I think it's kind of neat, beautiful, in fact, right? And so let's go ahead. I'm going to go ahead and yeah, here we go. On to the next operon. This is going to be the trip operon, which is repressible as opposed to inducible. Okay, here, the short version of this is that tryptophan, the amino acid, will turn off the genes that are needed to make tryptophan. Okay, so kind of makes good sense. If we have plenty of tryptophan around, then we don't need to make more, right? Tryptophan is one of these it's not an enzyme. So tryptophan is what again? Well, no, you don't know this because you're not a major student. Okay, but in BIO110 you would be learning that tryptophan is one of the 20 amino acids. Okay. It's the amino acid that's there in abundance in turkey meat. And it's also one of the starting points for the production of dopamine and the neurotransmitters that make you sleepy. And so one of the things you'll hear from many biology teachers is that after a big Thanksgiving dinner, you get sleepy, in part because your body is metabolizing the tryptophan, and it's converting the tryptophan into the neurotransmitters that cause you to doze off. Right? So yeah, there's a chemical reason for getting sleepy after eating a lot of turkey. It has nothing to do with your gluttony. It's basically biology, right? So tryptophan it's also relatively not a very common one. Turkey is one of those rare sources of tryptophan. Warm milk is another source of tryptophan. So if you drink a lot of warm milk, this might be another reason why it causes you to become sleepy. I don't know if being warm is going to make any difference, but theoretically it shouldn't. There's probably some other reason why warm milk is more soothing than cold milk. Okay, but in any case, tryptophan it's an amino acid that if it's not present, okay, if you have no tryptophan, in your diet, then E. coli, which is basically relying on the food that you're eating, your E. coli needs to make tryptophan. Because E. coli are very resourceful. You can't live without eating a source of tryptophan, right? You have to get whatever tryptophan you need from your diet. If you don't have it, then you're basically malnourished. You're missing that essential amino acid. E. coli don't need to have tryptophan in their diet. They're able to be a lot more flexible where they grow because they are able to make their own tryptophan, um, which is one of the 20 amino acids required to make polypeptides, right? And so again, we're going to come back to the operon model. Here's the control region. Here's the operator. Here's the promoter. Okay. And here are the genes that are required to make tryptophan, right? So we've got 1, 2, 3, 4. There are actually 5 genes total, or 5 genes in the TRP operon, right? And so once we get past this point, we'll be making all 5 enzymes that are required together in order to make the tryptophan amino acid, okay? And so if there's no tryptophan, okay, we'll skip right to the chase right here. When is on good for this? Well, when there's no tryptophan, right? And so you get this repressor. You get this repressor molecule drawn in the same color. If there's a repressor here that is going to bind or not to bind to the to its ligand, the ligand here will be the amino acid tryptophan. The tryptophan is going to bind to the ligand. Now, should this repressor, should this molecule right here, should this be tending to bind to the operator when tryptophan is present or when tryptophan is absent? Well, when tryptophan is absent, right? Okay. So if you have a ligand, no tryptophan, so the repressor with no tryptophan bound, this is going to not bind to operator, which would then allow the RNA polymerase to go right along here, start producing this mRNA transcript. By the time we get out over here, we'll have a nice long messenger RNA transcript, which will be read by the ribosomes. Ribosomes will be reading this. We'll be producing really long polypeptide. Let's see, do the polypeptide green, which will then be cleaved into 5 little fragments. And these 5 fragments will then turn into the enzymes that are required to make tryptophan. So basically, on is good when no tryptophan is absent. So the repressor is sitting there. It's not going to bind to the operator until tryptophan comes in, right? So when is off wanted? Okay. Yeah, when tryptophan is abundant. Tryptophan comes in, it binds to the repressor. Then the repressor changes its shape. It attaches, oops, not there. It attaches to the operator, and at that point, the operator will be functionally blocking transcription because it is keeping the promoter from reaching the parts of the DNA of the bacterium that have the information required to make the tryptophan enzyme producing genes. Yeah? I thought so. Let's take a look at what the book has to say. Yeah, okay, so this is exactly what we were talking about before. Do the annotate thing. Okay. Here is the, this is the repressor here. This is the RNA polymerase, And so the operator is here, and the operator is basically, in this case, it's actually saying that the repressor is blocking the RNA polymerase from actually binding the promoter. Yeah. That may or may not be true. I'm not entirely sure what the current status of our understanding is, because we might have learned that relatively recently, and I might be reflecting some old school learning by saying that the RNA polymerase might actually bind, and it would be blocked to from moving forward. But in either case, we're not going to get any transcription if the repressor is bound to the operator. The allolactose comes in, binds to the operator, and that's when this, binds to the repressor. That's what makes the repressor inactive, allowing the transcription to proceed. Scroll up a ways, and we get to the distinction between a repressible operon and an inducible operon. Even though they're kind of both using the same basic mechanism. You've got an operator and a repressor. And I guess the difference is, well, in the case of the lac operon, the lactose ligand is what causes the repressor to become inactive. And in contrast, in the case of the trip operon, the ligand attaching to the tryptophan is going to cause the repressor to become active and bind to the operator region, right? And again, the rationale for that is completely transparent. You just have to think about it for more than a couple of seconds, and you'll realize that all this makes sense. You don't have to remember any of this. This is not a memory thing. This is one of those things where you might impress a few people if you can explain it to them, because it sounds like you're explaining something that's got a whole bunch of moving parts. But the reality is, all you have to do is to think through this and say, yeah, it has to happen this way because we need to turn on the trip operon when trip is not abundant, right? So when trip is not abundant, that's got to be the inactive repressor. When trip is abundant, that's when we want to turn it off. That's why it's got to work the way that we described it. Okay. You look at the lac operon, it's the opposite way around. When lactose is abundant, that's when we need to turn the repressor off. We need to deactivate. We need to de-repress the operon. And so the repressor, in the case of the lac operon, is going to be different in the sense that it becomes inactive when it binds to the lactose. Right? Let's take a look at where we are here. I think I've covered everything. Operon structure. Yeah, we have several gene products all read off as a single RNA transcript. Then we've got translation, and then after translation, the gene products are separated. Got another enzyme that snips them into 3 little bits. Right? The genes in the case of the operon are related in function. They have to go together. Without one, you don't need the others. And the rules for determining whether or not the transcription is going to be turned on and turned off. Okay, that's determined at the control region. Specifically for us, we're focusing on that operator, which either allows the promoter to bind and move across or not, right? Then we detail the lac operon. And be able to explain to me, what is lactose and why does E. coli need to use lactose sometimes but not others, right? And these are enzymes. What is needed in order for E. coli to use lactose? These are enzymes encoded in the lac operon. Those are the 3 gene products, right? And so you've got the promoter and operator structure of the control region. You've got the RNA polymerase that's required for transcription. You've got the repressor, which will bind to the operator. And then you've got the ligand, the lactose, which is going to be attaching to the repressor and changing it from being an active repressor to an inactive repressor. And again, this is what we talked about at the very end. We want this repressor to be active. In other words, the whole thing is turned off. When? Okay, so when do we want the repressor of the lac operon to be inactive? When lactose is present or when lactose is absent? Answer that. Okay, and now that you got that one out of the way, we want the repressor to fall off the operator when lactose is abundant. Yeah. And then we talked about the trip operon. Remember, there's kind of like a context here. So you want to know, what does the trip operon do and makes tryptophan? So you want to know what tryptophan is. And why would we want to turn the tryptophan, why we want to turn off the transcription of the trip operon when trip is abundant. Okay. And yeah, so there's a lot of pieces here, but think of this as fun to think about. Okay, and you'll have your notes in the exam, so you shouldn't need to worry about this. Make yourself a little chart, work out some notes that will clearly guide you towards an understanding of the lac operon and the trip operon, and that'll be a fun project for you as you prepare for the exam. Okay, so if you thought this one was complex, just wait until we get to eukaryotic genome structure, because our cells, we've got a ton more genes than bacteria, and most of them are turned off. In contrast to the bacteria, where a lot of them are turned off, right? We're talking 20,000 genes, of which we don't need nearly that many. We need a few hundred genes in any given cell, right? And so for most cells, most of the genes are going to be turned off. And how do we make that happen? And how do we turn on the genes that actually are needed? And how do we make this different for your skin cell versus your muscle cell, right? Because the skin cell has to have different genes turned on from the muscle cell, and we need to be able to turn on the genes that are appropriate in the skin cell and not in the muscle cell. And so all of those kinds of questions are the ones we'll be talking about in the next video. Okay,